welcome. Today, we're going to take a tour of a super busy metropolis, the human cell. Seriously, thinking of it like a city with a government, factories, highways is the best way to make these high-yield USMLE concepts really stick. Okay, so here's our map for the tour today. We're going to start at City Hall at the cell cycle, swing by the industrial district to see how proteins are made, check out the city's infrastructure, and finally, see what happens when the building materials themselves are faulty. First stop, City Hall. This is where all the big decisions are made about growth and division. We're talking, of course, about the cell cycle. Okay, so here are the phases of the cell cycle. But honestly, the most important part for your exam are these checkpoints, especially that G1/S restriction point. You can think of them like permit offices. They will halt all new construction if the blueprints aren't perfect, and that's what prevents uncontrolled growth or cancer. So who's actually running this place? Well, the engines pushing the cycle forward are these cyclin CDK complexes. But what's really high yield for your boards are the city officials that hit the brakes, the tumor suppressors, P53 and RB. P53 is like the ultimate safety inspector. It senses DNA damage and slams the brakes on the whole process. And RB, it's the super strict gatekeeper at that G1 to S transition. You are not passing until RB gives the okay. All right, so we've seen how the city's governed. Now let's head over to the industrial district and see how this city actually builds stuff. We're going to check out its manufacturing and shipping departments. In our cell city, you got two main types of factories. First, there's the rough ER. It's called rough because it's covered in ribosomes, and its job is to pump out proteins that are going to be shipped out of the cell. Then you have the smooth ER. No ribosomes here. This is more of a specialty plant, handling things like making steroids and detoxifying poisons. And check this out. This shows you the whole logistics network in action. Proteins get made in the ER, and then they're sent over to the Golgi apparatus. The Golgi is basically the city's central post office. It sorts everything, packages it up, and ships it to its final destination. So what happens when the post office makes a huge mistake? Well, that's I-cell disease. The Golgi is supposed to slap this mannose 6-phosphate tag on certain enzymes. It's like a zip code that says deliver to the lysosome. But in I-cell disease, that doesn't happen. The enzymes get shipped out of the cell by mistake, and all the cellular garbage just piles up inside. It's like a citywide sanitation strike, and it's devastating. Okay, let's move on from manufacturing and look at the city's infrastructure. We're talking about the roads, the bridges, the public transit, all the stuff that gives the cell its shape and lets things move around. The cell's got this whole internal scaffolding called the cytoskeleton, made of three main filaments. But for long-distance travel, the thing you really need to know about are the microtubules. These are the highways of the cell. They're the tracks that all the cargo moves on. So what are we looking at here? This is a cross-section of a cilium, one of those little whip-like tails used for movement, like clearing mucus out of your lungs. And see that pattern, that 9+2 arrangement of microtubules? That's a classic buzzword for the boards. It's powered by these little motor proteins called dynein, which act like tiny engines, making the whole thing move. So what happens when those little dynein motors just break? Well, you get a condition called primary ciliary dyskinesia, specifically Cartaginer syndrome. Without working cilia, you can't clear your sinuses, sperm can't swim, and weirdly enough, during development, your organs can end up on the wrong side of your body. For your exam, you absolutely have to know this classic triad. It's a direct result of one tiny broken motor. We've spent a lot of time inside the cell. Now let's step outside and look at the actual construction material the cell uses to build everything around it. We're talking about collagen. It's like the steel rebar of the human body. Okay, making collagen is a whole assembly line process. You do not need to memorize every single step. But there are two steps here that are ridiculously high yield because they need specific cofactors. Look at step 2, hydroxylation. This step absolutely requires vitamin C. No vitamin C, you get weak collagen, and that's scurvy. Now look at the very last step, number 7, cross-linking. This is what gives collagen its incredible strength, and it depends on an enzyme that needs copper to work. Now, there are a bunch of collagen types, but for step 1, you have got to know these 4. Type I is in bone. Type 2 is in cartilage, see the 2. Type 3 is found in stretchy things like arteries, and type 4 is on the floor. It makes up the basement membrane. As you can see, a defect in any one of these leads to a very specific and very testable disease. So we know how to build it. But what happens when that rebar, that construction material, is faulty from the start? Let's look at some urban disasters caused by bad collagen. First up, osteogenesis imperfecta. The problem here is faulty type 1 collagen. The rebar in the bones is weak, so they fracture incredibly easily. To lock this in for your exam, you have to know the beat mnemonic: brittle bones, eyes that are blue, meaning blue sclera, bad teeth, and ear problems like hearing loss. Now, Ehlers-Danlos is another collagen problem, but it looks totally different. Here, you see things like hypermobile, super flexible joints and really stretchy, fragile skin. There are a few types, but the one you really have to worry about is the vascular type, which is a defect in type 3 collagen. It's the most severe because it can lead to arteries rupturing. And remember that collagen assembly line we talked about? Well, Menkes disease is what happens when that very last step, the final welding, fails. It's a genetic problem where you can't absorb copper. No copper means the cross-linking enzyme doesn't work. The result is weak collagen. And the classic sign you'll see on an exam is brittle, kinky hair. All right, the tour is over. Time to see what you learned. Let's do a couple of board-style questions. Think of it as your city planning exam. Okay, take a second, pause, and think about this one. We've got a kid with a major waste disposal problem. The garbage disposal enzymes are ending up in the blood, not where they're supposed to be. Where in that postal system did things go wrong?
Yep, the answer is mannose 6-phosphate. This is a textbook classic presentation of eye cell disease. The Golgi post office completely failed to add that M6P zip code. So the enzymes got secreted out of the cell, leaving the lysosomes empty and useless.
All right, here's the last one. Think about this city's transit system. You've got 3 problems in 3 totally different parts of the body. What single, tiny piece of cellular machinery could possibly link all of them together? And you got it. The answer is dynein. This is the classic triad for Carter-Jenner syndrome, and it's all caused by that faulty dynein motor protein. One protein failure paralyzes cilia all over the body, leading to all those symptoms. See? Understanding these pathways is absolutely key to crushing your exam. So what cellular process are you going to go investigate next?